Okay, well, thank you very much for the introduction. Also, thank you to the organizers for allowing me to talk to you today. I'm going to talk today about some of my postdoctoral work. So, I think we have a problem in computational biology, and that problem is one of choice. This is a fairly typical microarray analysis pipeline. So we start off with quantitation of intensity spots, move on to background correction, assess spot quality, and then to perform inter-array and intra-array normalization. So when we implement a pipeline like this, we have a lot of choices to make. For quantitation, there are several algorithms we can use, 3. When we do background correction, there are 11. Normalization, over 20 algorithms for both inter and intra-array normalization, which kind of tallies up to an enormous possible number of pipeline configurations that we can use. Okay, this problem isn't limited to microarray analysis. If we look at single nucleotide variant calling, we see again in the choice of alignment algorithm, we have several choices. When we choose the quality thresholds, we can vary our threshold according to our needs. At the pre-processing step, again, we have choices. There's a huge variety of SNV calling algorithms available. So when we perform this analysis again, we have a huge variety of choice. And why is this important? It's because these choices really matter. Like, these choices have a huge influence on our results. This is a paper that was published last year, and the authors showed that given different microarray pre-processing pipelines, the ability of microarray expression data to validate biomarkers varied enormously. So this data, these pre-processing methods really affect our results. So I'd really like to emphasize that. This is work that's ongoing in our lab currently. This is a benchmarking study of different copy number variant detection tools from whole genome sequencing data. So if we look at this heat map, this is each row represents a sample. Each column represents a different CNV calling algorithm. The color represents agreement with a gold standard kind of chip to call copy numbers. So as you can see, the variation data from the same algorithm is much more similar than data from the same sample, which is potentially cause for concern. So how would I like to address this problem? I'd like to answer these questions. The first thing I'd like to answer is which steps in a pipeline are the most important. And what I mean by the most important, I mean if we alter that step, which step will have the most profound effect on our results. Secondly, I'd like to use that information to try and optimize the existing pipelines that we have. And thirdly, I'd like to look for nonlinear relationships between steps. Okay, so this is a general overview of our approach. So we start off with a pipeline that we want to analyze and some raw data. So this approach is generalizable. We then perform an exploration of the available parameter space. In some instances, this will be it'll be possible to explore every configuration exhaustively. In other situations, we have to sample from the possible configuration space. So for every parameter configuration that we generate, we can run the algorithm or the pipeline and generate an output. The next step is to evaluate that output, usually against a gold standard data set, which gives us a metric of the algorithm's performance. We can then apply multi-level linear modeling to try and identify the important parameters and work on identifying the good targets for optimization and to try and predict good parameter sets. Okay, so that's the overview. So now I'm going to talk about how we apply this to specific data. The first data set we looked at was a single nucleotide variant calling pipeline. We used the GATK, which is a very widely used SNV calling pipeline. The very interesting thing about GATK, it's very, very highly parameterized. So the input is some aligned reads or a BAM file. We were working with 10 tumor normal pairs from the CPC gene prostate cancer study. So the first step of GATK is pre-processing. This pre-processing has 8 binary parameters and 15 numeric parameters. Okay, the second step is a probabilistic model used for the SNV calling. This has 5 numeric parameters. The third and final step is a variant recalibration. And this has 7 numeric parameters and 13 binary parameters. Typically, these numeric parameters are things like thresholds, priors on Bayesian distributions. Binary parameters are pieces of information that we can choose or not to give to the algorithm when it builds models. Okay, so even just ternarizing these numeric parameters and these binary parameters, we have somewhere in the order of 800 billion possible pipeline configurations for this algorithm. Okay, obviously this is impossible to evaluate exhaustively. So what we did, we sampled 400,000 configurations from this space. And obviously I'm sure you can understand this represents a very, very significant CPU investment in this study. Okay, what we did then is evaluated the results from each configuration. So each configuration gives us a set of SNV calls. We then evaluate that against a gold standard SNP chip. We calculated the area under the curve, so the area under the receiver operator characteristic curve, which gives us a kind of balance between sensitivity and specificity. Finally, we performed linear modeling to estimate the percent variance explained. In this context, percent variance explained is a proxy of parameter importance. Okay, so the next slide just shows you the kind of the size and the complexity of the space that we're dealing with. Okay, so essentially every column is a parameter configuration. Every row is a parameter. I'm not going to go into too much detail. I appreciate it's a lot to see. But just to show the complexity and the size of the space. So we have 400,000 columns and 48 rows. Okay, the configurations are sorted left to right by their score, so how well they perform. So the worst performing metrics are on the worst performing configurations are on the left, and the best performing configurations are on the right. Okay, so let's drill down into that data a little bit. So as you can see, the percent variance explained is focused in the second phase, the probabilistic calling model. These are the 20 most important parameters that we detected according to percent variance explained. So in the left panel, let me just walk you through this graph a little bit. So in the left panel, we see a heat map, and each column represents a decile. So what I did, I divided the parameter configurations into 10 deciles and put the worst performing 10% of configurations on the left and the top performing 10% of configurations on the right. So each column represents a decile of configurations. The color in the heat map represents the mean value of the parameter, the mean normalized value of the parameter in that instance. So what we can see here is that in the worst 10% of parameter configurations, high min-based quality score is highly enriched. So we see, if we look at the left column, we see parameter settings that we really should be avoiding. And in the right-hand column, we see parameter sets that might be good targets if we want to try and optimize our parameter sets. Okay, so just to give you an idea, min-based quality score basically applies a hard threshold in the second phase, and it throws away any reads that don't meet that threshold. So by setting that threshold too high, we end up throwing away the majority of our reads, and consequently lose any ability to make accurate SNV calls. And that results in a drop of the area under the curve from approximately 0.97, which is what the default parameters give us, to somewhere in the order of 0.5. So it's no surprise that this parameter has such a profound effect on the percent variance explained. Okay, so remember, this is a generalizable method. So this is genomic SNV calling data. So now I'm going to talk about mRNA expression data. So we have a nanostring data set available from the MAQC study, which is the microarray quality control study, so we have raw nanostring counts from various different samples at various different sites. We also have qPCR gold standard expression data. In this pipeline, there are 4 steps, as you can see on the color bars. So each column represents a separate parameter configuration that's encoded by the colors. Each row represents a gene. And the color is the fold from the expected expression count as defined by the PCR gold standard. So as you can see, blue is too high and green is too low. So the first thing that leaps out of this from this data is these 2 columns that are highlighted in red. Okay, something is going dramatically wrong here. And if we look, these correspond to the VSN step of the final normalization, which is variant stabilizing normalization. So you can see that looking at this graph, this VSN step seems to be the root cause of the majority of the variation present in this data set. And sure enough, when we apply percent variance explained, we see that the final normalization step takes the majority of the variance in the data set. So if we're looking to work on a nanostring pipeline, the first thing we should be looking at doing is paying really close attention to that final normalization step. This is what that tells us. So from microarray, from nanostring mRNA expression data, we can move on to, so from expression, transcriptomics to proteomics, we have another data set available, which is mass spectrometry peptide detection. We have some liver samples and we have some lung samples from mouse, and we know which protein should be occurring in the liver, which protein should be occurring in the lung. So we do a differential testing experiment to detect which peptides are present in one and not the other, and vice versa. So here we have a similar three-step pipeline. Again, the columns represent a unique pipeline configuration. They're sorted left to right by quality. Each row is a peptide that was detected, and the purple represents the p-value of the differential test. So again, the first thing that leaps out are these two columns on the right. We see different patterns associated with this column. Okay, and that's interesting to us. That corresponds to the normalization step of the mass spectrometry pipeline. Okay, so when we look at the percent variance explained, the majority of variance is explained by the normalization step. So when we perform a mass spectrometry experiment, we shouldn't be so concerned about the search method used. There are different methods you can use in the first step to compare your spectra to your database to identify which peptide you're detecting. So that is not a large cause of variance using different search methods. However, the normalization approach is a very large cause of variation in the data set. Okay, so I'd just like to conclude by saying that the choices that we make are really important when we talk about parameter configurations. These can make a huge, huge difference to our data sets, and we should pay close attention to it. And we should try and explore this space and understand this space a little bit better. We can identify the important parameters using very simple linear modeling techniques, and that kind of gives us information about which steps we should be looking at more closely. However, identifying optima is challenging. Predicting the optimal parameter set is very difficult. Predicting data is very easy, but validating that data on an independent set is difficult, just because there still remains a large amount of unexplained variance in these models. So this is challenging. In the future, at the moment, I'm actually working on how many configurations we need to evaluate before we can be confident about these assertions we're making about our pipelines. Okay, so I'll just leave you on that note. Thank you very much. I'd like to thank particularly everyone in the Boutros lab, especially the people in Bold who've done lots of work for this study, the genomics team at OICR, CPC Gene, Project for the Data, and funders, Prostate Cancer Canada, the Ontario Institute for Cancer Research, and Genome Canada. Thank you.
Josh?
Great talk. I had a question, two, actually unrelated. One is, have you thought about looking at combinations of parameters when you're analyzing, how much variance they're explaining? And is there a way, this is not my second question, is there a way to sample more? It seems like you're still undersampling the space, right, by 2 million-fold, if I got the calculation correct. Can you do an online type of sampling if you recognize that there are redundancies among the parameters and kind of sample in a smarter way?
Sure. The sampling approach is something that's, the results are probably sensitive to the sampling approach. In this context, it's very difficult to look for interactions, purely because we're probably underpowered to look for interactions in a data set as complex as the SNV calling pipeline that I showed you. But in the other data, we have another data set that we're looking at, which is chip data, so chromoimmunoprecipitation data. And in there, we certainly have power to look for interactions between different steps of the pipeline. I'm not sure if that answers your question.
Yeah, and the other one's easy. Is this available? Can other people use it? Is it plug and play into a pipeline?
Sure, I mean, I have code available. I don't have a tool, but the code itself is very, very straightforward. Thank you.
Hi. So, are there cases where the default parameters provided in these pipelines are particularly poor, or that seems Seems to be sort of a
Well,
for general people.
sure, that's a difficult question. I think in the GATK is the only pipeline that we analyze that really has a bona fide default sets. And certainly I have found parameter sets that I think are superior to that default set. There are a couple of examples where the second phase is perhaps overly stringent in what it throws out, and the third step can be actually trusted to do a good job with a filter. So I found parameter sets that I think are better, but it's very difficult to show that they are conclusively better in that case. Yeah, I think.
Probably depends on depends on the sample too, right?
Yeah, exactly. So what I actually do, I evaluate based on the delta between the default set and the configuration that we're examining.
What's your opinion about, let's say I'm using any of these pipelines and then I'm feeding it into yet another pipeline of analysis. Would you suggest that in that case I just try to find sort of pick the most variant pipeline spaces, or are you able to define some kind of okay, if I don't care about false positives, go for these parameters with GHK, but if I care about false negatives, go for these parameters?
Sure.
I mean,
yeah, that's certainly something that I looked at a lot. The advantage of sort of doing the sampling a priori is that we can use, so I have all the calls in a database, and it's very simple to generate a new metric. So if you're interested in weighting false positives more heavily than false negatives, that can certainly be taken into account in the metric design. So we can certainly do that.
thank you.
So I have one question myself. So it seems to me that you're using sort of the fixed grid partitioning for analyzing a parameter space. I mean, there's a lot of methods, Gibbs sampling, adaptive partitioning, that you could use to save a lot of computation and explore the more relevant part of the parameter space.
Sure.
Have you thought about that?
Yeah, the problem is with a sampling regime such as that is that you need to, once you have your genotype calls, you need to evaluate the metric at that point. And then you need to use that information to sample a new space, which is kind of a lot more computation intensive than the parallelization that we have now. And also you have the problem where, in response to the last question, you can't adjust your metric on the fly.